Hi, I'm Jun Kim. I'm a postdoctoral scholar in Julian Sage's lab at Stanford University. And today, I want to talk about targeting cancer metabolism to develop new therapeutic. In physics, the first law of thermodynamics states that energy can neither be created nor destroyed. So to create and maintain life, an organism has to derive energy from somewhere. Cellular metabolism is a chemical process that derives and consumes energy. It is a fundamental process that maintains life at both cellular and organismal levels. This also applies for cancer cells. The main characteristic of cancer is uncontrolled cell division, eventually taking over a patient's body. Again, energy can neither be created nor destroyed, so cancer cells require constant and increased supply of energy to maintain rapid cell divisions. This requirement gives cancer cells unique metabolic characteristics that can maximize cellular proliferation. For example, it is well known that cancer cells show increased dependence on glucose to maximize carbon supplies. But when the flow of energy is interrupted, cancer cells become extremely vulnerable and often succumb to death. This is analogous to a bad driver who is out of control and accelerates until the last drop of gas instead of slowing down or refilling the tank. Such vulnerability of cancer cells has been leveraged since early on. Even before first cancer mutations were discovered, metabolic inhibitors or antimetabolites were tested as cancer drugs. And this really revolutionized cancer treatment in the 1950s. Many of these drugs are still being used as first-line therapies for different cancer types, and this makes antimetabolites the oldest and one of the most effective class of drugs that are out there. After more than half a century since the approval of the first antimetabolite, almost all of the approved drugs still target nucleic acids. But nucleic acids are just one of the 4 classes of macromolecules that are required by cancer cells. Different cancer cells rewire circulation of these macromolecules differently, complicating interpretation of experimental data. But the recent revolution in high-throughput technologies has revealed metabolic characteristics of cancer with unprecedented resolution. And I am using these technologies to reveal novel vulnerability in cancer. There are many genes regulating metabolic pathways, and not all cancer cells are in the same metabolic state. So we really need a way to analyze many genes at the single cell level to get a comprehensive view on cancer metabolism. And to do this, I am using single-cell RNA sequencing and mass cytometry. Single-cell RNA sequencing is a method that quantifies gene expression, or RNA, in individual cells. Mass cytometry is a method that quantifies proteins in individual cells. And together, these technologies can observe metabolic regulation at both RNA and protein levels in single cell. To inhibit the molecules that are critical for cancer progression, I'm also engineering enzymes to chemically remove these metabolites, and I believe the enzymes can be developed into new cancer drugs. Efficacy of cancer therapy remains low, and cancer patients are faced with limited options. My dream is to cure cancer in every patient, but I also know that it's not possible at this time. By conducting metabolic analysis cell by cell and sample by sample, I'm devising strategies to treat a wide group of patients and combining cutting-edge high-throughput technologies such as single-cell RNA sequencing and mass cytometry. And with these, I can start to dissect metabolic requirement in individual cells and identify vulnerability in cancer as whole. Through using these technologies and by developing candidate drugs, I hope to contribute to both science and clinical management of cancer.